Welcome to our review on the spread of communicable diseases. So first thing we need to know is how we actually have these diseases that spread from person to person. Now, if we consider the main routes of infection, then we tend to see these occurring through cuts in the skin because we've lost that physical barrier, through the digestive system where you've eaten food that's contaminated with something, through the respiratory system where you've breathed something in, or through the reproductive system. So to give you an idea about one of the key pathways, we've got this thing called droplet infection. So in the top right there, you can see the lovely image of what happens when you sneeze without covering your mouth and nose. So you can see that lovely splatter pattern of all of these little droplets of mucus, and each of those individual droplets could contain those pathogens that lead to diseases. So when someone coughs or sneezes in your vicinity, then all of those little droplets there are just being emitted into the air. If you inhale those droplets, then you would catch that disease, okay? So this is what's known as droplet infection. So droplets of mucus that contain the pathogens are emitted from the mouth and nose when someone coughs and sneezes, and then someone else inhales it to catch the disease. We've mentioned the fact that plants also have these communicable diseases. Now, the method by which these plant pathogens are going to spread from one plant to another is slightly different. So what we find is one of the main ways that our plant pathogens can spread is through soil and water. But that's not the only ways. They can also be spread by things called vectors. Now, a vector is just something that's going to carry the pathogen from one plant to another. So this would be things like an insect that feeds on one plant and then goes to another one and then spreads the pathogen in that manner. We could see it as a direct contact of sap from an infected plant with a healthy one, and this could be down to agricultural damage. So if you're using little secateurs to take cuttings and you don't wipe them between, you can spread those diseases from plant to plant. And it could also be down to the feeding of animals. If they feed on an infected plant and then go to an uninfected, we can spread there. The last method is airborne means. So things like the wind can spread those fungal spores from one plant to another. So once our pathogen has actually infected a plant, it doesn't cause disease instantly. What we actually have is this period of time between that point of infection and the actual symptoms of the disease or feeling ill. And that's known as the incubation period. So during that incubation period, what we see is the pathogens are reproducing. And as they do this, then they're going to cause damage to the cells that they're actually invading. Other pathogens may also release toxins, which will have negative effects on the host organism as well. If we come on to consider how these different microorganisms are then going to replicate, we see different situations whether we're talking about a bacteria or a virus. If we think about bacteria first of all, then they replicate through a process called binary fission, which is basically mitosis. And what we actually see is that they experience something called exponential growth. So in the bottom left, you can see the diagram of binary fission. We start with one bacteria, that's then going to split into two, those two split into four, and so on. Now, if you think about how that's going to see a population increase, the graph on the middle there actually gives you the idea. And this is an exponential growth graph. So it starts off really low, but then it increases very, very quickly. So that's what we mean by exponential growth. Viruses are a different scenario though, because a virus can't replicate on its own. For a virus to reproduce, it has to take over the host cells and then it uses them to generate new viruses. And the way it does that is that it inserts its genes into the nucleus of the host cell. It then tells the cell to basically use all of its replication machinery to make new viruses. And then once there's enough of them, they literally burst out of the cell, destroying it, and then those viruses, off they go, and then infect other cells. One thing that scientists do to actually track the spread of these communicable diseases is they study what's called the incidence of a disease. Now, when we talk about incidence of a disease, quite simply, that's the rate at which new cases occur in a population over a certain period of time. So we might see something like 5,000 new cases per week. So that would obviously be talking about the fact that within our given population of a country, there were 5,000 new cases of Ebola occurring each week, for example. And when we study the incidence of a disease, then we can actually have a look and see if there are any particular patterns. Does it link into particular seasons? Is there a particular insect or animal available at the time that acts as a vector? So it gives us a greater understanding and therefore gives us the ability to hopefully come up with ways to prevent the spread of that disease. Hopefully at the end of this video you can now describe how communicable diseases can be spread between animals and between plants, and you can explain how scientists actually monitor those different diseases and their spreads.